Heart development. The earliest sign of the development of the heart is the appearance of angioblastic cords that become canalized to form the primitive heart tubes. Folding of the embryo plays a key role in heart development. In this longitudinal section, after folding of the head of the embryo, the primordial heart, along with the septum transversum and the pericardial cavity, migrate onto the ventral surface of the embryo. During transverse folding of the embryo, the bilateral early heart tubes fuse to form a single heart tube. The developing heart tube is initially suspended by the dorsal mesocardium within the pericardial cavity. As the heart tube continues to develop, multiple components can be identified. The thick external layer, the myocardium, is formed from splanchnic mesoderm surrounding the pericardial cavity. The innermost layer, the endocardium, is derived from the original endothelium of the heart tube. A gelatinous connective tissue layer, known as cardiac jelly, separates the myocardium and endocardium. By the beginning of week 4, the tubular heart has started to elongate, and a series of alternating dilations and constrictions can be seen. These are the primitive ventricles, atria, and aortic sac. The multiple layers of the heart wall can also be seen. The venous and arterial ends of this primordial heart are anchored in place by the septum transversum caudally and the pharyngeal arches cranially. During the 4th week, the developing heart undergoes a right-handed or dextral looping, which results in the apex pointing left. And the atrium and sinus venosus are relocated dorsal to the ventricles and outflow region. This same looping and complex repositioning can be seen in a lateral view as well. The development of the heart requires contributions from specific cell populations and is influenced by more than 500 genes. Lineage tracing studies suggest that progenitor cells of the pharyngeal mesoderm anterior to the heart field are one such population, and that waves of these cells contribute first to the ventricular myocardium and the outflow tract. A second wave, or secondary heart field, contributes to the rapid growth and elongation and provides cells to the myocardium of the left ventricle and the anterior pole of the heart. This same series of developmental steps relating to progenitor cells is shown in this lateral view and on close-up. By the end of the 4th week, atrioventricular, or AV endocardial cushions are formed from the dorsal and ventral walls of the AV canal. In this side view, a simplified version of the cellular processes involved is shown. The endocardial cushions develop from specialized cardiac jelly or extracellular matrix. This requires an inductive signal from the AV canal myocardium and an epithelial mesenchymal transformation of the cells. These transformed cells then invade the extracellular matrix. During the latter part of the 5th week, the primordial heart components progress in development with specified morphologic changes. In this diagram, the relative position of the AV canals and associated endocardial cushions change as a result of the expansion of the myocardial tissues in the inner curvature of the heart. Simultaneously, the endocardial cushions fuse, resulting in distinct right and left AV canals, generated from a single canal. The subdivision of the AV canal is shown in more detail in these images. The endocardial cushions expand in size and approach one another ventrally and dorsally. As a result, distinct right and left AV canals form, and the fused endocardial cushions for the AV septum divide the two canals. The mitral and tricuspid valves control blood flow between the left and right atria and left and right ventricles, respectively. The development of these valves begins with proliferation of mesenchymal tissues surrounding the AV canals and the remodeling of the primordial ventricles. The valves develop from subendocardial tissue as swellings, which become hollowed out to form the valve cusps, 2 in the mitral valve and 3 in the tricuspid valve. Both valves are prevented from prolapsing into the atria by the tension of the chordae tendineae. Tissue that contributes to the atria arises from several locations. The right atrium is derived from the sinus venosus, and for this reason it has a smooth surface lining. The rest of the anterior atrial wall and the right auricle derive from the primordial atrium and have a trabeculated or rough surface. Most of the left atrial wall is smooth because it is formed by the connection of the pulmonary vein to the left atrium. As these veins become connected, there are initially 2, then 4 openings that can be seen within the left atrium. In the right atrium, the crista terminalis is the demarcation line between the smooth and rough wall portions. The development of the interatrial septum is best demonstrated by viewing mid-sagittal and coronal sections simultaneously. The endocardial cushions can be seen as well as the primordial atria and ventricles. Initially, a crescent-shaped membrane, the septum primum, begins to grow toward the endocardial cushions. This membrane originates from a pool of cells in the mesodermal cap of the developing atria. In the right atrium, the crista terminalis is the demarcation line between the smooth and rough wall portions. As the membrane is formed, the opening between its edge and the endocardial cushions, the ostium primum, decreases in size. This opening allows blood to be shunted from the right to the left atrium. As the membrane approaches and fuses with the endocardial cushions, perforations and then a complete opening develop in the more central portion of the septum primum. This opening, the foramen secundum, results from apoptosis and shunts blood between the right and left atria. Finally, a second but partial membrane, the septum secundum, begins to grow from the ventrocranial wall of the right atrium, immediately adjacent to the septum primum. The remaining opening of the septum secundum is known as the oval foramen. Together with the septum secundum, the oval foramen provides a pathway for the blood in the right atria to shunt toward the left atria. Partitioning of the primordial ventricle begins with myocytes from the left and right ventricles, which form a ridge or septum in the floor of the ventricle. This muscular interventricular septum grows toward the endocardial cushion both by proliferation of the myocytes in the septum and dilation of the ventricles. The resulting interventricular foramen is closed at the end of the 7th week by a membrane. Formation of the membranous interventricular septum will be described at the end of this animation. The outflow region of the heart requires the migration of neural crest cells through the primordial pharynx and pharyngeal arches. This migration provides the cells necessary for the formation of the bulbar and truncal ridges. In these diagrams, the primordial cardiac outflow regions can be clearly seen. The blue and yellow ribbons represent the conotruncal ridges that form from the migrated neural crest cells in the walls of the outflow region. In cross-section at various levels, these ridges can be seen to have a spiral orientation within the lumen. The conotruncal ridges fuse, resulting in the formation of two separate arterial channels, the aorta and the pulmonary trunk. For this reason, the pulmonary trunk is oriented spirally around the aorta. The most distal portion of the outflow tract, the aortic sac, is partitioned by the development of an aorticopulmonary septum. The location of this septum is coordinated with that of the conotruncal ridges. The semilunar valves begin to form when the partitioning of the distal portion of the outflow tract, the truncus arteriosus, is almost completed. Here we see the separation of the two arterial channels. The semilunar valves each develop from three swellings of subendocardial tissue around the orifices of the aorta and pulmonary trunk. These swellings form the right, left, and either anterior or posterior valve leaflets. In this sagittal view, the swellings can be seen undergoing a hollowing out process that leads to reshaping and formation of the thick-walled cusps of the valves. The formation of the muscular interventricular septum provides most of the required tissue for the separation of the right and left ventricles. However, a small interventricular foramen remains open. A small membranous septum is formed to provide final closure. This membranous interventricular septum is comprised of tissue from three sources: the right bulbar ridge, the left bulbar ridge, and the right side of the endocardial cushion.